all. This is Dr. Mobeen Sayed from DrBeen.com. Welcome to one more show. The discussion today is a few set of news that are really interesting to look at. A quick showdown or a quick summary of the news is the following. One, there are some researchers who have figured out proteins that can be virus neutralizing, SARS-CoV-2 neutralizing. So these proteins are different from antibodies that are neutralizing. And so we'll talk about that. Secondly, I want to talk about the UVC rays or the Ultraviolet-C rays and the lamps that are available for these. And finally, we'll talk about CDC, although I don't feel much inclined to talk a lot about CDC anymore, given the chaos that they have. But still, it is interesting to see that they have put the airborne transmission back onto their page. So at least it's interesting to see what they're doing. So with this, let's start. So the very first thing here is DrBeen.com. What I have done is, or my team has done, is that we have created this page, and I have the link in the description. This page is for the COVID-19 videos. So far we have edited and uploaded about 79 or so videos out of 184. And so if you see here, these videos are out there, plus you can actually earn CMEs with them as well. So this is available. The link is in the description. So with this, let's start. And once again, welcome everyone. The discussion, first part of the discussion, here is the reference or the link. Computer designed proteins may protect against coronavirus. So this is like brand name proteins or designer proteins. So let's look at how these work. What they've done is the following. So this project is led by Dr. David Baker. The objective for them was to create proteins, so not antibodies, proteins that can bind to the receptor binding domain of the coronavirus or the spike protein of the coronavirus. So that was their basic purpose. And what they did was they employed two ways to take care of this virus or this binding protein thing. Number one, what they did was, this is very smart. So check this out here. Over here, this is a cell, correct? And the cell has the ACE2 receptors on it or ACE2 enzymes on it. What they did was they took parts of the ACE2 enzyme structure. So ACE2 enzyme itself is a protein as well. So they took some part of that protein, incorporated that part in another protein. So imagine this red part here is actually a piece of the ACE2 enzyme. So then they took that and incorporated that into another protein and made a new protein that has a unit that looks like ACE2 enzyme to the spike protein. And then they tried to see if the coronavirus, if the SARS-CoV-2 will bind with that protein. This is very similar to once I had a friend who had joined our YouTube lecture. His name is Dr. Vasaris, Alexander Vasaris. He's from Greece. And he had said that why can't we have decoy ACE2 receptors, or why can't we have fake ACE2 receptors, which when injected in the body can go and bind with the spike proteins of the virus. Excuse me. So this is that similar thing. So here they have produced a designer protein which looks like ACE2 receptor, and then that protein is supposed to bind with the SARS-CoV-2's spike protein and neutralize it. It is not an antibody. So I'll explain what is the benefit of it being not an antibody. Then what they did was, in a second way, or the second method, they took existing designer proteins. So there are thousands of millions of protein structures that we have in our labs. And you can just make new protein structures all the time. It's nothing. These are amino acids. This is like alphabets of the English language or any other language. And then you just string them together and make words and sentences. Similarly, you can take amino acids and put them together, and the resultant thing will be a protein. So there were thousands and thousands of proteins present in their database. So what they did was, with the computer's help, they went over the designs of those proteins and kind of figured out that what proteins may be able to bind with the SARS-CoV-2. And again, receptor binding domain of the spike protein of the SARS-CoV-2. So imagine that these are the proteins, and these are many, many proteins. I just made 4 or 5 here. And these proteins have various shapes to them. And then what they did was, they looked at those shapes and asked the computer to tell them that what is the best possibility, which shape has the best possibility of binding with the RBD of the spike protein. This is like you going into your tool shed and then seeing what tool can be connected with what kind of whatever bolt or nut or something. So here they found a couple of proteins that were interesting. They called them LCB1 and LCB2. So they originally found 10,000 proteins. Out of those 10,000, then they found 2 of the proteins that were able to bind with the RBD of the spike protein. So if you see here, very fascinating. I love this one. So first, they incorporated a segment of the ACE2 receptor into the small protein. The researchers used a protein design tool that they developed called Rosetta Blueprint Builder. So that is one. Secondly, they took the existing mini proteins from scratch, and then they kind of saw what can be a good protein that can bind, and they found 10,000 such proteins. Then they filtered them and they tweaked them and they tried to use them with the SARS-CoV-2, and then SARS-CoV-2 or maybe a simulation of the spike protein, and then they ended up with 2 proteins called candidates LCB1 and LCB2. So then what they did was, they brought those candidates, and if you see here, this is the second protein, and these proteins are now bound to the spike protein of the coronavirus, and that is how they know that this would bind and neutralize this RBD. Then they tested them in the following way. They had lab-grown human cells. So imagine these blue guys are the human cells. So they put them in a lab experimental dish, and then what they did was, all these red over here are the coronaviruses, or possibly some virus with a similar SARS-CoV-2 type spike protein. Then they put those designer proteins, or mini proteins as they call them, on top of this. And so here, these blue dots, if I show here, if I zoom in, these blue dots are the binding proteins that were then supposed to bind with the spike protein. And what they found was that none of the cells in this experiment got infected. That means that the proteins they had made were functional and they were neutralizing, and so they had been successful. They of course say that more testing is needed, and I believe that, because we got to figure out those proteins when injected in the human body, where are they going to go and what are they going to do, and other than binding with a coronavirus, are they going to bind with other things, are they going to cause other issues? But remember, we eat various proteins all the time. Although when we eat a protein, it is not necessary that the protein itself ends up in our body. The proteins are digested and broken down into smaller pieces, which are the amino acids, and then we can pull those amino acids and then use those. Imagine that amino acids are like bricks that make proteins. So then we take those amino acids to our blood and in our body, and then we use those amino acids or bricks or alphabets and then reassemble into the proteins that we need. Here we will be injecting proteins from outside, so they can have their own issue. So now, how can these be different? So they were not very much clear about how these are going to be different from the antibodies. But let's say for a second that here we have an antibody, and here we have a binding protein. What is going to be the basic difference between them? Look, what happens is, it is possible that both of them are able to connect with the SARS-CoV-2's spike protein. So this is a spike protein, and they both are able to connect with the spike proteins and neutralize them. However, the antibody structure is formed in a way And our body knows that to be in a way that it can actually do more than just neutralizing the pathogen. For example, antibodies can get connected on macrophages on the FC portion or with FC receptors. So there, one part of the antibody can actually bind with the macrophages, and then this whole complex of antibody and the macrophage can be internalized or phagocytosed and then digested and acted upon. That's one possibility. Then antibodies they have areas on their constant region that activate the complement, and complement in turn then activates the immune system and helps the immune system. Remember, we talked about complement in the past, that complement can make holes in the pathogens surfaces, plus complement can activate the immune system. They can cause chemotaxis of the immune cells like neutrophils and others to come to the area of infection or area of destruction and cause the effect. So these proteins that are now connected with the SARS-CoV-2, they do not have the function of an antibody. So that may be good or that may be bad. Why is it good? For example, these proteins are not going to activate the phagocytosis. So the result of that is that these are not going to be triggering the immune system or the macrophages, and then those macrophages are not going to create tumor interleukin 1 and 2 and cytokines and all that. Or macrophage activation syndrome may not occur because of this. Secondly, these proteins will not activate complement. Complement in turn causes local damage plus causes chemotaxis of the immune system cells to come over. So these proteins will not amplify, in simpler terms, these will not amplify the immune system. They would just go in and bind to the virus and just neutralize it. The question that I have not been able to answer yet, and again, I think even the researchers would say that, yeah, we need to do those testings. The question that I cannot answer is, what will happen to this complex? So the virus and the protein, this complex, what would happen? How would that be cleared out? Will this still be phagocytosed by macrophages or dendritic cells? They would still digest them. Will this complex go into the renal system and maybe get stuck in the filtration system over there? Will this just keep running around in the body? Will this get deposited in various tissues and then cause our immune system to react to both of those things and cause immune responses? So I do not know about those, but these are the kind of questions that we will have to see answered before we can say that these proteins are going to be useful. Our body generally is able to look at proteins, work with them, degrade them, and then throw them out or digest them as well. So we make proteins all the time. Even antibodies themselves are proteins. So we make them and then we break them, and we are okay with that. However, what would happen with these new proteins are not known. It still is a fascinating thing. I loved it. So that is one. The other discussion that I wanted to do today is about the ultraviolet-C radiation. So there is a news circulating out there that UVC 222 nanometer wavelength lamps are safer, relatively safer for human skin and eyes, and should be used. So I wanted to make sure that we can look at it together. So let's look at some of this together over here. So first of all, this is FDA's UVC light lamp guidance. So I have this link in the description. I would request that you please go and check this out. Here they have given a lots of important notes. The important note for the UVC that we should know is that it has to fall on the virus directly. So for example, if there is a surface, let's say this is a porous surface, and the light, just like right now, the lights are falling on the surface, but there, if you see there's a hole in it, this is actually the pen holder for my digital pen. So there is a hole in this thing. And if the viruses are in it and the light cannot fall on them directly, then those viruses will not be killed. So one of the important thing with this is that the light has to fall on the virus directly. That is one. Secondly, we have to be away from that place because it can irritate our skin and cause even damage to our eyes. So this site over here, dose of radiation, direct exposure of the radiation, and then what are various kinds of UVC lamps. There are some lamps that produce ozone, and then some that do not, and some has mercury and some do not, and so on. And then how should we use them? What wavelength is interesting? So I am just going to give you a visual tour of the same discussion that they have over there. So let's check that out. First of all, the Ultraviolet-C lamps are known to be disinfectants, and they can disinfect air. That means they can kill some pathogens and viruses that are in the air, water, and non-porous surfaces. Why do they say non-porous surfaces? Because the porous surfaces have pores in them, which are microscopic indentations in which the virus may be sitting, and the light may not reach in those holes, microscopic holes, and kill the virus. So non-porous surfaces are the one that can actually be used, that can use the ultraviolet rays. So now, how does it work? There are multiple ways to use these lamps, and I'm going to show you a lamp here. This is a lamp on Amazon. And if you actually Google or search on Amazon and you say UVC light lamp, you would come up with many kinds of lamps. Most of those are in this range. For example, this lamp that I have here is 253.7 nanometer. And you can find 222 nanometer wavelength lamps. However, please be careful. I searched lots of lamps here. I looked at their specifications. I looked at their reviews, and then I looked at what kind of caution is accompanying the lamp. And if you see here, it says, so I just want to read the caution here. Because the UV rays will hurt eyes and skin, so please don't look at the working bulb. So don't be present there. People, pet, plant can't be exposed to the light, must left room when lamp work. I think they did some Google Translate. This may be coming from a different country. Must leave room when the lamp is working. Liquids such as perfumes and wines should not be exposed to the light, as strong UV rays may cause chemicals to deteriorate or decay. Don't put brightly colored clothes. So basically, textile dye can actually be decayed or damaged by the UVC light. So that is why they're saying don't put brightly colored clothes, toys under the light for a long time, because a strong oxidation of UV ray may cause slight fading. They actually, in the reviews, people talk about fading of the furniture as well. So if I have, let's say, this table over here and I have the UVC lamp, it is possible that my table can become discolored or my furniture can become discolored. Keep the bulb out of reach of children. So fine, that is fine. So going back to the discussion here, the best way to use a UVC lamp is to actually install it with the air system. So air ducting system. So put it in a duct. So when it is added to a duct and the air is passing from there, it would disinfect the air without having the exposure towards humans. So that is one way. So if you see here, this is a lamp here, and this is installed with the air ducting system, maybe at the HEPA filter area, and then that air is circulating. So this is one way, that is air disinfection. Then it can actually be put on water as well. So this is my rather simpler way of showing it. So let's say this is the UVC lamp here, and this lamp, and down here is the water flowing, and the lamp is emitting UVC rays, and the water that is flowing underneath is getting disinfected. Now, what I could not find, and I think this can be found if somebody did more research, is that how deep can these rays penetrate? So what would be the water's surface, water's depth that will become disinfected by what strength of the lamp? So that is the piece of information that I do not have for you over here. Then surfaces. So if you see here, this is, let's say, a surface, a stretcher or something, or a table, or surfaces at home, and this is the lamp here. So the lamp would emit the rays, and that would cause the surfaces to be disinfected. Once again, the surfaces have to be non-porous. I looked around today in my home to see that how many pieces of furniture do I have that are not porous. And I think almost everything is porous. There is wood or there is metal, very few very shiny and polished surfaces that may not have pores. And then, so some considerations for the UVC. Number one, it cannot inactivate hidden viruses. So if you are shining the light on soil, for example, it would not work. If you're shining it on porous surfaces, it would not work. If you're shining it on a layer of clothes, then the top layer may have some benefit, but the clothes underneath will not have any benefit. So it only inactivates when it is directly shining on the virus. Then have not been thoroughly tested. So FDA says that we know that it works with SARS-CoV. We also know that it works with some other pathogens, but we actually do not know we don't have enough data for SARS-CoV-2. Even the 222 nanometer wavelength that is in the news nowadays, still they say that we don't have enough data to say, yes, go ahead and do it, and what should be the dose, and what should be the radiations, excuse me, duration, and so on. Then please realize it is harmful for skin and eyes. The 254 nanometer is more harmful than 222 nanometer, which is less harmful. So that is all the riot nowadays, that, hey, we have 222 nanometer that we can use. And if you see here, they would say, check this out here, the good surface, which I do not know what is good surface, 5 minutes once. So how to use good surface, maybe they mean non-porous surface. 5 minutes once. Put goods, also goods surface. So goods meaning packaging and other things. Put goods within 4 feet of the light, such as cups, bottles, toothbrushes, shavers, cell phones, keyboards, clothes. Then small spaces, 5 minutes once. For example, shoe racks and cabinets and drawers. Small room, 15 minutes. That is, for example, toilets and such. Larger rooms, 15 to 60 minutes. So that is the kind of amount of radiation needed. And I saw in the reviews that many people were saying that, well, I put it in a large room and I had a UVC detector and it did not detect the light at a distance, which tells me that there is a possibility of a perception of safety while the lamps may not work that well or that far. And we might think that, all right, they're working and the surfaces are clean, and they may not be clean. For example, if we try to use them in schools or other places. So there is a danger of having a bad impression that the area is clean while it may not be clean. So back here, so let me show you one more thing. I have been doing some research for you. There are commercial products as well. So these are the UVCs on Amazon, and there are many of them. And I have no relationship or no interest with any of those. I just Googled them and I went over to them and saw which one looked better. So here there is an Apache safe space solution. So here they have various kinds of commercial UVC lamps. For example, if you see here, this top one, this lamp would draw air in it, then shine UVC light on it and kind of clean the air and then throw it out. Then down here, this is another industrial grade lamp, which would be probably attached to the ducting system. And then it would pull the air, work on it, and then emit that. Or it can be probably placed in a room and it pulls the air in it, cleans it, and then throws it out. I also wanted to show you the prices. So for example, this lamp, which pulls the air, cleans it, and throws it out, this was $1,850. But again, this is a commercial lamp. Then down here, this is handheld lamp. So here if you see here, the person is holding it in their hand and they are kind of shining it on various surfaces and various places, or maybe even in the room. So these were $560 to $1,000. And then this is another lamp. If you see here, this is another lamp. Then they had this camera as well that can look at a crowd of people and detect the higher temperature and then tell who may have fever. Anyways, This was also an interesting site I found. So then length of exposure matters, dose of radiation matters. Some lamps produce ozone or ozone, whichever way it is pronounced. So that is an important part. And I saw on Amazon that some people had complaint about some lamps that said that, hey, the lamp said it will not emit ozone, but I had some sort of an ozone meter and I detected ozone. So, and the FDA site says that as well, that be careful, some of these lamps would produce ozone, and ozone is then irritant to our respiratory system, and it would cause irritation and possibly damage to the respiratory system. Then some lamps have mercury in them. So when they are broken, then mercury is toxic. So taking care of the broken lamp and making sure that the mercury does not cause too much of a damage is also an issue to keep in mind. So back here with the CDC, so there are various kinds of lamps here: low pressure mercury lamp, excimer lamp for UVC 222 nanometer wavelength, pulse xenon lamps, light emitting diodes, and so on. So I would recommend that please look at it. It may be interesting to use, but at the same time, please make sure that you are safe from its hazardous side. And finally, the last topic for today is once again CDC. So I think that it's not too much fun to continue to talk about CDC. They are just lost. But once again, remember, this is their how does it spread? And I'm using the Wayback Machine or the web archive to see their previous pages and the new page. So this is a page from August 5. And here there was no airborne or aerosol transmission. Most of this was primarily droplet and maybe fomites. I actually think that they are wrong on saying maybe fomites. Fomites are the surfaces or areas where somebody has touched and those surfaces have become contaminated. Maybe somebody's snot or the nasal secretion or the spit or other secretion or the sweat, or well, sweat doesn't have it. But if they have, let's say, their spit or the nasal secretion put on some surface, that surface may have viruses. So they're not very much clear on that, and they say it may happen. Primarily, they said droplet. Then remember that they once again went from droplet to a possibility of airborne, and this is the one where they went to airborne. This was September 20, and then they said, oh, we made a mistake. This was a draft that got published. We were not supposed to do this, and we take it back. And so they said no airborne. And now if you go back here, now they acknowledge, or they have added, that respiratory droplets is one way of transmission, then airborne transmission is another way. This time, there is a slightly more careful way of articulating their message. And so they have said that, hey, airborne is when the bigger droplet becomes smaller and then it stays in the environment. And then if it stays there for 30 minutes, then it would cause an issue. Otherwise, it would not cause an issue. So to me, it seems like it's essentially, if you read it, it almost looks like they are saying it can cause the transmission, but it won't cause the transmission. So they're trying to now stand on both sides. So if you see here, there is evidence that under certain conditions, people with COVID-19 seem to have infected others who were more than 6 feet away. This is the important part here. When there is an airborne transmission or aerosol transmission, then the tiny droplet, or the nucleus we call them, can actually go to a farther distance. Or it can just stay suspended, and even if the person has gone and somebody else comes in, that suspension could have SARS-CoV-2 in it. So that is the discussion that they had done. So this is the discussion for today. I wanted to go over some of these things with you. I also wanted to today discuss the vaccines, and then I wanted to discuss a letter in New England Journal of Medicine where they had said that the current administration has not been able to protect the people. So they put their weight on what should happen. However, I think we don't have time today, and if you like, we can talk about it tomorrow. The other is vaccine, that Moderna and Pfizer's vaccines have some people who had severe reactions. but they say that those reactions or those events only occurred for a day or so, and then they recovered, but these events did occur. So I'll talk more in detail tomorrow if possible. So here we are. Thank you very much for today. I'm just going to look at some of the comments here, and then we will close and we'll meet each other tomorrow. And once again, if you are looking to support my work, there is a link in the description for donations. And then please like, subscribe, and share as well. Yeah, so there is a comment about the blood-brain barrier. So these are called the stellate cells. James says, fascinating science. Thank you. We are very blessed. Thank you very much, James, and stay safe as well. There is a question from Barbara. Dr. Bean, would you respond to Muhammad bin Shaida's serious issue? No oxygen and they need ideas, help in Tunisia. What is this? France, Lorraine, McDonald, Farmer, Barbara Warren. It seems a concentrator is the only option. I got dead people are thrown in the garbage in here. So Muhammad bin Shaida, you have my phone number as well, and you have sent me some emails too. We can talk through that as well. It seems like an issue that I may or may not be able to talk about. Cool. So guys, thank you very much. Once again, please like, subscribe, and share. Stay healthy and happy, and I would see you tomorrow.